Unit 8, Molecular Genetics. This is 8.4, Mutations. What is a mutation? Well, okay. Mutations are a change in the DNA. That's it. That's all it is. Change in the DNA. Well, that's a big deal, right? Because the change in the DNA is going to result in a change in the messenger RNA, which is going to result in a change in the protein, which is going to result in the change of the function of that protein. So it could be a bad thing. Not necessarily, though. We're going to get to that. And so you see here, the original copy is supposed to have green. The mutant copy is going to have red, whatever red and green stand for in this particular case. And that's a mutation. There are different kinds of mutations. Let's talk about them. There are point mutations. A point mutation is when a single base has been changed. The original is supposed to be UUA, but if you have a CUA, that's different. So that's a mutation. However, there can be three different outcomes of these point mutations. The first one is called a silent mutation. What is a silent mutation? Well, UUA codes for leucine, which is an amino acid. CUA also codes for leucine, an amino acid. Is it a mutation? Yes. Does it cause a problem? No, because the amino acid is the same. Protein's going to have the same shape, same function, no problem. Still a mutation, though. That's a silent mutation. The next one's called a missense mutation. Well, a missense mutation changes the amino acid, right? So instead of UUA, now I have GUA, which codes for valine, which is not leucine. So that's going to change the shape of that amino acid. That's going to change the function of that amino acid. This is probably going to cause an issue in the function of that protein. So that's a missense mutation. And then lastly is a nonsense mutation. Nonsense mutation causes an early stop codon. UUA codes for leucine, but UAA codes for a stop codon. So the protein stops. It's supposed to have 10,000 amino acids. Instead, it only has 7. That's going to cause an issue in the function of that protein. And so these are 3-point mutations. A single base has been changed, 3 different outcomes. Moving on. The next is called a frameshift. A frameshift results from an insertion or a deletion of a base, a single base, a codon, a bunch. It could be any number. And so what does that mean? So first of all, the reading frame is groups of 3, right? We've established that. Those are called codons. And it's going to cause that frame to shift over. Well, let's look at the original up here. Here you have AUG. What's AUG? It's the start codon, right? And it's going to just keep on going. Everything's good. Protein folds up. Hey, this is what I'm supposed to have. Everything is great. Fantastic. Here's the DNA. Here's the RNA. Here's the amino acids. This is transcription, translation. Everything's great. No one's having a problem. Well, down here, we insert a C into the amino acid, or a G, or both, whatever. There's an insertion, right? Well, it's not replacing something. Notice it's inserting it. And so the rest of the DNA is having to shift over in order to make room. Well, when it shifts over, you no longer have whatever this is, CAA. After that, you have CAC, which doesn't code for GLN, it codes for histine. And then you no longer have tyrosine here, you have isoleucine, right? And you no longer have glutamic acid, you have arginine. And so notice what happens after the insertion. Everything is broken. And in fact, this causes an early stop codon, which is just not good. And so a frameshift causes a problem for the protein after the insertion or deletion. So if it was deleted, it would just be scooted back this way. Same kind of problem is going to occur from that. And so very important concept, the idea of frameshift, it's going to break everything after it completely. There are some mutations that affect the whole chromosome. All right, so we're going to look at these. So insertion, deletion, this is easy. This is when the idea that a whole section of a chromosome is either inserted or deleted into another chromosome. So here you have this piece is inserted into that one, or a piece is taken out, right? So that can happen, insertion or deletion. This is going to affect multiple genes. This is going to affect multiple layers. Inversion, instead of ABC, these are supposed to be genes. They're CBA. Well, that's going to cause the genes to be read backwards, which is going to cause some issues, obviously, right? So if a gene is inverted, it's going to cause some problems. Duplication is where a section of the DNA becomes duplicated. We're going to talk about some instances in a moment where this is not necessarily a bad thing. Could be, not necessarily, right? So translocation, This is where just a piece of the chromosome breaks off and relocates or swaps. This can happen naturally, or it does happen naturally, obviously, but this can be beneficial or detrimental also. We're going to talk about the difference between those two things. Moving on. Also, another thing that can affect the mutations if whether or not they're germline or somatic. What does that mean? Well, somatic is like any cell, like a body cell. So let's say you have a mutation on a particular body cell, right? And that mutation is going to affect a particular part of the body. Whereas if there's a mutation on, say, like a sperm cell or an egg cell, then that's going to affect every cell in that individual. So here's showing a mutation after fertilization. This is showing a mutation prior to fertilization. So when we say germline, anytime you see that word germline, it's talking about either sperm or egg. And so if there's a mutation in the sperm or egg, every cell is affected, whereas if there's a mutation on any one of the body cells, only that particular area of the body is going to be affected by that mutation. Spontaneous mutation, these are typically mutations that occur during replication. So like there's just a problem with replication. There's some sort of error. There's 6 billion base pairs. Errors happen all the time. There's actually proteins that their whole job is to like find errors and cut them out. But that's not always going to be the case either. So some of these can go on. These are called spontaneous mutations. And there are other mutations that are caused by things called mutagens. Which mutagens is just something that causes a mutation. UV light is a great example of this. Other carcinogens like cigarette smoke is another good example of this. Can cause mutations in a particular area of the genome, which can cause any sort of problem to occur, right? Cancer is a great example. When we use the word carcinogens, we are talking about things that cause cancer, and cancer is generally caused from a mutation in the DNA. Are they harmful or beneficial? Depends on who you ask. So a great example of this is bacteria. So bacteria are generally non-resistant to antibiotics, right? Hence the word. However, because of mutations, some of those bacteria, they could have a resistance to a particular form of amino or to antibiotic. Well, if those bacteria are allowed to live, then, well, the new population is going to be mainly resistant. So those antibiotics are no longer going to be effective. And so this is a mutation. It's not as if the bacteria decided they were going to become resistant. It's just a random thing that occurred that caused resistance. Here's another example. So early fish did not have a jaw, right? This pre-nathosome means just pre-jaw fish. Jaw mouth is basically what that means. And so over the course of evolutionary history, what's happened is some of the genes of the jawline duplicated, which caused the jaw to have this different kind of structure. And you slowly, through the process of natural selection, slowly what happens is you have this jaw form that is different than the original because these genes duplicated. Now, some might think that, well, gene duplication could be a problem. Well, in this particular case, it's not. There's other examples of gene duplication happening in snakes. And so you have a salivary gland, then you have this other gland that becomes the venom gland, right, which has been a good thing for snakes. And so whether or not they're harmful or beneficial, that's the words that we'll use rather than good and bad to determine a gene's or a mutation's effectiveness. Does it cause the organism to become more fit? If it does, it's beneficial. If they lose fitness, then it would be considered harmful.